Okay, so today in this lecture we will discuss about the cell cycle, and specifically we are going to discuss about the mitosis. Before we move forward in this process of cell cycle, I want to clear some basic concepts about the chromosomes. So, for example, this is one chromosome and this is the centromere, and this chromosome has DNA. Okay, and the end of this portion is called the telomere. Now, and why I am discussing here is because during the cell cycle there is a replication of the DNA. So now if the DNA is replicated, then what will happen? Again, there is a one chromosome or you can say the two sister chromatid. So you can decide the number of chromosomes by counting the number of centromeres. If suppose there is one centromere, then you will have one chromosomes and like that. So number of chromosomes are equal to number of centromere, or the vice versa. If there are n number of centromeres, you can say you can define the number of chromosomes. Okay, if suppose I am making the diagram like this, and again the number of centromere is one centromere, so there will be one chromosome, or you can say three chromatids. So that is one thing. So you can define the number of chromosomes by counting the number of centromeres. Number one thing. Then coming to the homologous and the non-homologous chromosomes. Non-homologous chromosomes. We have 23. Human beings have 23 pair of chromosomes. Okay, or you can say the 46 chromosomes. One copy of the chromosome you are getting from mother and one copy of the chromosome you are getting from father. Homologous chromosomes have the same set of genes. For example, for the eye color gene, one copy you are getting from the mother for the eye color and one copy you are getting from the father for the eye color, and both of these genes will be located on the same number of chromosome. Let's say if this is the chromosome number 1, and just for the sake of example, suppose the eye color is present, the gene for the eye color is present on the chromosome number 1. So you will get one copy from your mother. Let's say this is your mother maternal chromosome, this is your paternal chromosome. So one copy of this eye color you will get from your maternal chromosome and one copy from your paternal side. And these genes will be located on the chromosome number 1 only. So the location of these genes are going to be same. So this is the homologous chromosome because they have the same set of genes. The eye color genes are present on the chromosome number 1. So they are the homologous chromosome. Now the non-homologous chromosomes. Suppose this is a chromosome number 13. This chromosome will have different set of genes that will not code for the eye color, that may code for some other color, let's say skin color or something else. So they will have the different set of genes. Then chromosome number 1. So they are chromosome number 1 and chromosome number 13. They are non-homologous chromosomes because they have different set of genes. So this is another thing which you, and X and Y, as you can say, X and Y are also non-homologous chromosomes. For the sake of the knowledge, I can tell you that the X chromosomes contain approximately more than 1000 number of genes, while the Y chromosome has about 300 or 400 genes, and that is basically responsible for the gonad formation. And that is why X chromosome inactivation occurs in females. Okay, because females have two X chromosome, males have X and Y chromosome. So males are lacking one X chromosomes. X chromosomes are involved in many processes that are required for the development or for the growth of the life. So in case of the female, there are two X chromosomes. So you will be having the two copies of genes, while the males will have the single copy of gene, and therefore there is a compensation in the females. One chromosome will become inactive, and that you can see when the lady is pregnant, doctor can see this X chromosomes in the form of bar body, and when they are seeing the bar body in the ultrasonography, they will tell that the child is female. They can tell you whether the child is male or the female. Okay, so X and Y are the non-homologous chromosomes because they have the different set of genes. X chromosome have genes which are required for the many processes of life, while the Y chromosomes have genes that are required only for the gonad development in males. And since we are talking about the cell cycle, I want to tell you about the cell division. On the basis of cell division, cells are divided into three main types. One is the labile cells. Labile cells basically they divide continuously. For example, hematopoietic stem cells, skin cells, GI tract cells, they need to divide. Okay, because they have a limited period of time. For example, RBCs, the lifespan of RBC is just 120 days. So if they will not divide, there will be anemia. So they continuously divide. Now, the another cells are stable cells. Stable cells divide whenever there is a requirement or there is an stimulus. If you are putting these cells under some stimulus, for example, in the presence of certain growth factors or other kind of, if there is any injury or something like that, then these cells will divide. And examples are kidney cells, liver cells. Then there are certain cells which do not divide at all. They are known as the permanent cells, or you can say the amitotic cells. They do not divide. For example, your brain cells. Then skeletal cells, which which help in the movement processes. Then the third one is the cardiac cells. So these are the three kind of cells. Now we are coming to the mitosis. In the mitosis, basically what happened, there are two growth phases, G1 and G2. There is one phase which is the S phase, where the DNA replication occurs, and then there is a mitotic phase. These three together are known as interphase. And in the mitotic phase, you have the prophase, metaphase, anaphase, then telophase, PMAT basically, followed by the cytokinesis. And you will have from one cell, you are going to have two cells. Okay, number of chromosomes will be going to same in these cells. Okay, now what happened in the G1 phase? And there are certain checkpoints which I am not going to discuss in this lecture. I'll make a separate lecture for that. So what happened in the G1 phase? In the G1 phase, the cell is preparing itself so that its DNA can replicate. And now, when the cell at the end of cytokinesis, you are getting two cells. And the if suppose this particular cell has one mitochondria, this particular cell will also have one mitochondria, and this is also going to have one mitochondria. So basically what happened, and similarly for the other things also, ribosomes, like nucleus, everything needs to be doubled, and this doubling occurs in the G1 phase. So here what happened in the G1 phase? Organelles, they are going to be doubled. If there are any thymine dimers, that needs to be removed. And as you know, for the DNA replication, you need certain enzymes that also need to be there during the G1 phase. Once the cell is sure that everything is ready, then only it will enter in the S phase. So the another things are certain enzymes, transcription factors. The cell will prepare everything here so that the cell now can enter in the S phase. What happened in the S phase now? In the S phase, what happened? DNA is going to be replicated. So here you can see, if suppose this is your G1 phase, and in the G1 phase you have 46 chromosomes. Now the cell is coming to the S phase and DNA is replicated. Now in the S phase also you have the 46 chromosomes, because number of centromere is only one. 46 chromosomes, or you can say 92 chromatids. And because DNA always replicate in the semi-conservative form, so here there will be the old copy of DNA. And one is going to be the, let's say, the new copy of DNA. Okay, this is your S phase. Now cell will enter, once the DNA is replicated, cell will enter in the G2 phase. So what happened in the G2 phase now, which is again the growth phase? You know, the number of organelles, they are doubled. Okay, DNA, that is also doubled. So now the cytoplasm needs to be increased in amount, the liquid part, and that happened in the G2 phase. Otherwise what will happen, the cell will burst. If the size is same and you are doubling everything, the cell will burst. So this is your this is what happened in the G2 phase. Now we are coming to the mitotic phase, which is the fourth number of phase, which is the mitotic phase. So it has the prophase. In the prophase, basically what happened in the interphase, the chromosomes are like the thread like structure, like this. Now in the prophase they become condensed, they become like this. So first thing that happened is condensation of chromosome. Second thing what happened is laminin phosphorylation, or other proteins also, they phosphorylate. Once they phosphorylate, they activate certain proteases that degraded the nuclear membrane. Okay. And the third thing happened is you will start observing the microtubules organizing center, which is the centrioles. These are the three things which you can see in the form of diagram. If you want to see your, this is suppose your cell, the nuclear membrane will disintegrate, chromosomes will condensed, and you will start seeing the microtubules organizing center. This is your MTOC. Okay, so this is the prophase. Now coming to the chromatophase. You might have heard the term of karyotyping. Karyotyping is usually done either at the prometaphase or metaphase, and that is why I am discussing about the prometaphase here. In the prometaphase, what happened? You will start seeing the kinetochore. Suppose this is your chromosomes, so there will be a structure, hook-like structure, which is the kinetochore. And your centrioles or microtubules organizing center will start moving towards the pole of the cell. This is your microtubules organizing center. So there will be microtubules like that. Like that. Similarly, it will be the another chromosome, and here will be the another chromosome. Okay. So this is your basically we are saying this is your kinetochore. Okay, and these are the microtubules or spindles, you can say. And these two sister chromatids are attached to each other by a cohesin protein. Okay, so this is the prometaphase. Basically, what happened in the prometaphase, you will start observing uh, the presence of kinetochore. You will start observing the movement of the microtubules towards the pole. But whatever I have shown here, this happened when these all chromosomes are lined up with each other. This is the condition of metaphase. So what happened in the prometaphase? First we'll write about the prometaphase. You will start observing movement of the MTOC to poles, and the second thing, you will start observing the kinetochore, and this is the condition of metaphase. In the metaphase, the chromosomes will aligned up on the metaphasic plate. So let's say, suppose this is the metaphasic plate, let's say. The chromosomes are aligned up at the metaphase plate. In the cytogenetics, when the scientists are karyotyping your DNA chromosomes, they are arresting your cells either at the prometaphase or the metaphase. So whenever you are seeing the karyotyping, you will always see like this, the presence of chromatids, because you can see the there is one chromosomes but two chromatids. You will start like this. You can see the karyotyping like this. Okay, this is a prometaphase and metaphase. Now coming to the third point is anaphase. Anaphase, the chromosomes now will start moving away from each other with the help of certain motor proteins which will carry them. One is the dynein and another one is the kinesin. Okay. So with the help of these proteins, now the chromosomes, they will start moving away from each other. So let's say this is your centrioles and these are your microtubules. Now chromatids, sister chromatids will move away from each other. So here you can see in the anaphase and in the telophase there are 92 chromosomes and 92 chromatids. Why? Because in the metaphase, when they were like this, they were 46 chromosomes and 92 chromatids, but now they are moving away. So they are like this. Number of centromere is there in both the chromatids. So what will happen? There will be 92 chromosomes and 92 chromatids. Okay. When these two sister chromatids are getting apart from each other, because the number of centromeres, the centromere is present in every chromatid. You can see now this is a chromosome. This is not a chromatid, this is a chromosome. But there is a 92 chromosomes or 92 chromatid during the anaphase. Now these chromatids, the sister chromatids, they will move apart from each other in the anaphasic conditions, leaving the 92 chromosomes and the 92 chromatids inside the cell. Now coming to the telophase. Number one thing that will happen is chromosome decondensation. They will again become like thread-like structure. Number two, laminin dephosphorylation. Number three, nuclear membrane will form. Okay, so these are the major things that are going to be happen in the telophase. Okay, again in the telophase there will be 92 chromosomes and 92 chromatids. Then coming to the cytokinesis, where cell will divide and form two cells. One cell will divide and form two cells. Okay, and in the process of contraction, because if this is suppose your cell, the actin filaments will help in the contraction of the cell, and here this cell will have all the organelles, your mitochondria, your ribosomes, nucleus, chromosomes, which is 2n, and the similarly here also the nucleus, chromosomes 2n, organelles, everything will be there, and then the cell will divide, one cell will divide and form the 2n cells, two cells. So number of chromosomes, if here there are 46, at the end of cytokinesis you will have two cells having 46 chromosomes. So now you can understand, in the G1 phase there are 46 chromosomes and 46 chromatids, because in the G1 phase what is there, chromosome is like this. Now what happened in the S phase? When it became like this, because number of centromere is 1, so here again there will be 46 chromosomes and 92 chromatids, and the similar condition will happen in the G2 phase till the M phase. Till the M phase this is going to be happened. Okay, now what happened? M phase with the metaphase. Now in the anaphase and telophase, there will be 92 chromosomes and 92 chromatids. Why? Because these two sister chromatids will apart from each other. So what happened in the telophage? If suppose this is the sister chromatid, they will apart from each other. They two sister chromatid. Now the number of centromeres are 2, so number of chromosomes. These are not chromatids now. These are now chromosomes. The number of chromosomes will be 92 and number of chromatids will be 92 in the anaphase and telophase. And then coming to the cytokinesis, after the two cells are formed, each cell will have 46 chromosomes again and 46 chromatids, you can say. This is the second cell, it will also have the 46 chromosomes or 46 chromatids. Okay. Okay, so again in the G1 phase you will be having the 46 chromosomes and 46 chromatid, because this is the structure. In the S phase there is a duplication, so again because number of centromere is 1, the chromosomes are going to be 46 but there will be 92 chromatids. In the anaphase and telophase, these sister chromatids are moving away from each other, but each sister chromatid is having the centromere, so it will work as a chromosome. So there will be a 92 chromosome and a 92 chromatids in the anaphase and telophase. And at the end of cytokinesis, you will be having the 46 chromosome in one cell and 46 chromosome in the another cell. Or you can say the 46 chromatids in the one cell and 46 chromatids in the another cell. So this is regarding the cell cycle. Now you can study the cell cycle by using certain dye, which is the propadium iodide dye, that can only enter if the cells are dead. So whenever you are staining your cells with the dye, you need to fix them. And how you can fix them? By using the paraformaldehyde. So once you fix your cells with the paraformaldehyde, and after that you add the propadium iodide. So propadium iodide is a DNA binding dye. It will bind to the DNA. So in the G1 phase, the DNA content is less. In the S phase, there is a replication, and in the G2 phase, your replication is completed. So you will be getting like this. In the flow cytometry, this is the fluorescence, and this is the cell count. And this is the histogram which we are talking about. Okay, so fluorescence is highest in case of the G2 phase. Why? Because the DNA content is more in the G2 phase in comparison to the G1 and S phase. In the S phase, the DNA content is more than the G1 phase but less than the G2 phase. And in the G1 phase, the DNA content is less. So if you are staining your cells with the PI dye, and this is the fluorescence intensity, you can say. You can say the intensity is high in the G2 phase in comparison to the S phase. In the S phase, it is high in comparison to the G1 phase. Similarly, there is one more dye, which is the BrDU dye, bromodioxyuridine. That mimic the thymine. So whenever the DNA is replicating, it intercalate between them. So it actually stain the cells when they are in the S phase. So you can stain the live cells with them. Okay, that only replicate and that only stain the cells which are in the S phase. Why? Because in the S phase the DNA is replicating. When the DNA is replicating, that because BrDU mimic the thymine, which is the pyrimidine base of the DNA, it will intercalate between the base pairs and there will be the fluorescence. So only the S phase cells you can stain with the help of this dye. So that's all about this lecture, and I'll discuss about the cell cycle checkpoints in my next lecture. If you like my video, please subscribe my channel. Thank you very much.